thanks for being here. First and foremost, I want to thank folks that have helped out on this project with me. I'm a postdoctoral researcher at the Rubenstein Lab at University of Vermont, and I work primarily on foraging models and food web models of the benthic habitat in Lake Champlain. And so this project was done along with my advisor, Jason Stockwell, really great undergraduate research assistant who was with me last semester, Jonah Ballard, and Brian O'Malley, who is at USGS at the Tunisian station, who, as you will see, contributed most of the data for this project. So, and I'm talking about sculpin behavior today. So. As many of you probably know, sculpins are a really critical species in freshwater habitats writ large, and I've somehow turned off your slides. Okay, never mind. They are used in part as a sentinel species, given they have a fairly limited home range, and therefore they can serve as a pretty good indicator of the surrounding habitat. And they also serve as this really important mid-trophic link in a lot of freshwater aquatic food webs. So they're a forage fish that are eaten by a lot of the larger, more charismatic species in, say, Lake Champlain. But they also are one of the major vectors for moving benthic nutrients and biomass back up into the water column via their consumption. So they're completely benthic fish. They don't have swim bladders. They spend their time on the bottom, and they're primarily eating, invertebrates and the like in that habitat. And then when they get eaten, all that biomass gets moved up. Unfortunately, while we know they play this role, we don't have really great estimates about the kinds of foraging pressure they do exert in the benthic habitat. So we know they're eating these invertebrates, and we know they're mobilizing those resources, but we don't necessarily know to what degree. And that in part is because that's mediated by their behavior, and observing natural behavior is really, really hard. We can do some stuff in laboratory, but oftentimes those conditions can be somewhat limited. So fortunately, a few years ago, for a completely different reason, Brian and Jason were working on another project with mysis. Mysis being one of these invertebrates that is eaten by sculpin, and mysis migrate in the water column. So they're one of these major vectors for moving nutrients. And Jason and Brian were looking for sort of the presence of mysis in the benthic habitat. And to do so, they built these really cool steel camera rigs. These things are about, I think, 50, 60 kilograms, and they're equipped with large battery pack, infrared camera, and some LED and some infrared lasers as well to give you a sense of scale. And they were dropping these down in the main lake of Lake Champlain right here in this little red square that I have for 24 hours at a time. So you didn't have to leave a boat on site. You could drop these rigs down and go back and collect them, change the battery, and set them the next day. And of course, they were looking for mysis, but they found plenty of other species. We'll happen to come across the video. They got some yellow perch, they got some sea lamprey, and maybe most frequently, they captured videos of sculpin. So this is an example of what one of the video frames looks like, giving the scale on the bottom. Those two white points are the lasers that give you a sense of scale. And here we have a sculpin entering the frame. And the playback is going to be a little jumpy, it seems.
Oh, very good.
So that's an example. This is an example of a sculpin actively attacking prey targets on the benthic floor. And this was not a one-time occurrence, but rather there is a lot of video with sculpin in it. Brian was kind enough to send me a hard drive with all of the video he recorded on it. So far, we have pulled out and processed about 6.5 hours of video that has at least one sculpin in frame, although we did find up to 4 at a time. And in fact, Jonah is right now finishing pulling some of these videos off of the supercomputer cluster back at UVM for me, so that number is going to grow a little bit by the time we're done making this data set. But I'm really here to tell you about how the data set is made. So this all I'm going to show you is going to be based on these 6.5 hours. And instead of Jonah or I sitting down and painstakingly, frame by frame, going through and trying to measure movements and do that for this amount of data to characterize the movement of this species, we went another route. So we used a software package called Deep Lab Cut. This is an open source software package. You could go on your computer right now and start making one of these models with any video that you have. I was first introduced to it in my undergraduate, and it's primarily used in laboratory. So it was developed with laboratory animals in mind. And what it gives you is pose estimation. So it gives you positional and relative positional information of various body parts that you're interested in tracking on an animal. So instead of me, say, measuring hand by hand between each frame how far a fish is moving, I can instead train one of these models and have it detect the positions for me. So what we did is we tracked 6 positions on the sculpins in our video. To train one of these models, you first have to give it a set of ground truth observations. So Jonah and I sat down and for, I think, a little bit over 100 different still frames from the video, marked these 6 points on each fish we could see in the video. So we did tip of the nose, around the nape of the neck, around where the dorsal fin is, tip of the tail, and then both pectoral fins. And just to give you an example of what that, once you've trained this kind of pose estimation model, what that actually looks like, hopefully my video renders okay. This is one example of it tracking a fish. As you can see, our dorsal point was a little jumpy. I can talk more about which body parts ended up getting used in final analysis, but this was one version of the post-trained model. It ended up with an error of about 10 millimeters, which we found to be good enough to work with. I'll talk a little bit about how we also cleaned the data some, given potential gaps and the like. But we felt that this kind of model gave us a good insight into describing the behavior of these fish. So this is sort of what the raw trajectories look like, meaning I'm looking at cumulative distance traveled regardless of orientation over time in a video. Notably, you'll notice we had some videos with the same fish in frame for 10 minutes at a time. Sometimes they did not go very far. But what I want to point out really to you here is more about the character of these lines. Sculpin, again, they're benthic fish. They don't have swim bladders. When they're not swimming, they are on the ground. They are on the bottom. And this also plays into one of the major ways that they search for prey, specifically via the lateral line system. So oftentimes they're foraging at night. Oftentimes they're foraging in very deep waters where ambient light is not available. And instead they're relying on a sensory system that runs down the middle of the body that is incredibly sensitive to, say, vibrations or changes in pressure. And as a result, what you end up seeing are these stop and go movements. These are also known as saltatory movements, where there's a brief period of movement and then there's a period of rest. So we wanted to characterize this behavior, and we chose 3 measures in particular. We chose the distances that they're moving, the durations that they take to move those distances, which also will give you velocity, and then the periods of time they're spending at rest in between movements. So to get those measures, we first did a little processing of the data just to clean it up a little bit. We ran into a few issues that we had to smooth over. So the first one would be gaps in the data. This is when the model would not reliably say it could identify where the fish was. This was a little common in our video just because most of these videos come from about 60 meters down from sandy substrate. So sometimes when the fish moves, the substrate clouds, the video is occluded. And depending on the direction of the current and whatnot, you could lose the fish for a few frames. So we took some tolerable gaps. We defined a certain length of gap that we found tolerable to still use, and we would interpolate positions across those gaps from where it was last detected to where it was next detected. But larger gaps we omitted entirely, and we said we're not going to calculate anything about behavior in those gaps. The second thing we did is a little bit of smoothing using a weighted moving average, just to reduce some of that noise from that remaining 10 millimeters of error in the model. And then we computed all of our kinematic measures step by step. So between each individual frame, this is 30 frame per second video. There's a lot of frames. And then finally what we did is we were looking at trying to detect the difference between movement and rest depending on a threshold of velocity. And what the end product of all this looks like is something like this. So you're seeing the video on the right, which if I did my animations right should be somewhat lined up with the video in the middle, where blue denotes periods of movement and gray denotes periods of rest. And then obviously you can see that the velocity varies for those different movements. So now just a little bit to tell you about the results. The first thing we did just at base was to look at the body size distribution of what we observed. Notably, we can't tell if a fish re-enters the frame, so this isn't necessarily like indicative of the kind of distribution you would get from a trawl. Say if a fish leaves and then comes back, I can't guarantee to you if it's the same fish or not, so it's counted twice, even if it is the same fish. This is just to say, this is what the movement data is based off of. And these are our measures that we were looking at according to body size. So body size is on the bottom. We have rest durations, movement durations, and movement distances. Main point I want to point out to you here is there wasn't really a strong relationship between body size and the movement behavior of these fish. So what can we do with these data? I'm a modeler by trade, so I'm interested in actually using these to parameterize some models that I work with in my other time. Again, we're interested in the amount of foraging pressure these sculpin are putting on benthic invertebrates, perhaps to tell us something about how much of those invertebrates they may clear and therefore potentially mobilize into the water column. So 2 ways we're exploring this right now. One is in encounter rate models, which is to say, how frequently should we expect a foraging sculpin to detect targets in its environment? And we've been developing a model based on the saltatory movement I described in synthesis with this detection radius, or sort of the range at which one could reliably detect prey. This is something that's been experimentally in laboratory measured by Janssen back in the 90s, specifically focused on vibrating beads in sand in laboratory. And what we can do with that is then, say, predict encounter rates as maybe that detection radius changes, perhaps because of different substrates or habitat complexities and the like. And the second thing we're working on is looking at not the search component of predation, but the pursuit component. So as you saw in that first video, and as you're hopefully going to see in this video here, there's these strikes that happen when the sculpin are actually going for a prey target. You can usually tell it pretty clearly because they'll blow some of the substrate out after the strike occurs. And again, in this Janssen paper, that's also what was one of their focal points, was the ability to triangulate these tactile stimuli, approach them through a series of short jumps, and then attack them. And so we're hoping to look at not only predation pressure in search, but of course actually as realized in pursuit. So one of the preliminary things we've measured here is the actual distances at which strikes occur in our video. Strikes being given by a certain level of velocity, and that I have hand-checked visually, which is a bit of a pain, but I want to make sure I'm seeing the gill blow out. So what I'm showing here is that detection radius measured by Janssen is the orange line on the outside, and then I'm giving you a heat map of distances at which strikes occurred in the viridis here. So the red is going to be the mean in the middle. And as you can see, the point at which these fish are attacking prey is much, much smaller than the point at which they're detecting them. So something for us to look at and hopefully look at with this data set is not only the search behavior, but also maybe the approach behavior that happens from detection to strike. The problem being we can't actually see the prey targets because most of them are in faunal and in the substrate. So it's a little tricky, but it's something we're thinking about. And just in terms of future directions, some things to think about. The study was like, for lack of a better word, the study was opportunistic. Brian was really generous in sending me that hard drive. I actually haven't met Brian yet, so if he's here, I'm very excited to meet you this afternoon. But I think that this is actually a really powerful approach to observing natural behavior. These things are down there long enough that the local fauna can habituate to their presence. Usually after impact with the substrate, you could see fish within 30 minutes in these videos. So but there's plenty to do, right? So because this was opportunistic, we didn't necessarily look at behavior across different habitat types at different depths or things like that. So one obvious next step could be to look at how behavior changes in different habitat types. Specifically, I've pointed out the presence of mussels as a potential factor that may significantly affect these foraging strategies given their nature. And we have zebra mussels in Lake Champlain. Quaggas are oncoming, so even in these deeper waters, this will become more and more of the norm. And then, of course, this approach can readily apply to other species. So I have highlighted, of course, our friend the round goby, which also has yet to make it to Lake Champlain, but is at the doorstep. And to potentially look at the different kinds of foraging pressures that different species are exerting in the benthic habitat. For example, of course, one could also look at some of the larger benthic fish as well. There are plenty of methods for using these things in 3D too, although, of course, getting clear water reliably is not necessarily an easy thing either. But I think that these kinds of technologies, they're getting more and more common. Again, this was an open access model that anyone could use. And I really think that folks should be looking into them as a cost-effective way of not only looking at presence, size distributions, but also some more nuanced things such as behavior. So, yeah, thanks for listening. Thanks to folks at the Rubenstein Lab who have seen this for a while now, and to USGS, and to the Great Lakes Fisheries Commission for the funding. Thanks.
Yeah.